So structural proteins we'll probably not come back to in the course, so you need to get this slide in your head now. Can you tell me what the cytoskeleton is? So cyto means cells, right? And the skeleton of a cell. So I've already shown you that the cytoplasm inside a cell is really full of proteins. And quite a lot of those proteins, particularly in animal cells, are full of things like tubulin, which we saw in the previous slide, which form like a scaffolding inside the cell to hold it in its shape. Right? Plant cells have a cell wall and turgor to hold their shape. Fungal cells don't have very much that holds their shape because they're trying to work their way through things. So their shape is really determined by their medium that they live in. But animal cells have to hold shape, and they do it by building protein scaffolding inside the cell. That's called the cytoskeleton. Right? Hormones. We learn about these a little bit later in Unit 2, but hormones are chemical messengers in the blood. Many hormones are proteins. So insulin, glucagon, adrenaline, these are all proteins. They're made by the ribosome, laying down amino acids with a code from the nucleus, from the DNA. But some hormones are not proteins. They're manufactured by proteins. So the sex hormones, what we call the steroid hormones, these are made by proteins, but the proteins are enzymes that are breaking down cholesterol into different forms. So cholesterol is a kind of fatty molecule, and it's used to make steroid hormones because the proteins can break cholesterol down in various stages to different types of molecule. And so estrogen and testosterone are not proteins themselves, but they're made by proteins. Okay? But there are a few that are proteins. Insulin, for example, is a protein. Glucagon is a protein. Right? Adrenaline won't come up again in our learning. All right? They tend to be small so they can get around. Okay, so protein receptors are located on the surfaces of cells. Okay, so they're membrane-bound proteins. And they have binding sites that are specific to a single target, such as a hormone. So the ones that we learn about in National 5 are hormone receptors. Right? But there are other types of receptors. There's receptors for just signaling, so cells can send out very tiny molecules as signals, and that increases the concentration of something outside the cells. There are molecules in the cell surface that are sensitive to those. But what you have to remember about receptors is that the reception happens out on the outside of the cell. The hormone doesn't get into the cell. What happens is that when the hormone binds to this end of the cell, chemical changes happen in the receptor protein, and something maybe gets dropped off on the inside. There might be a phosphate group bound to the protein here, and the binding of the hormone out here causes that phosphate group to drop off. So when phosphate concentration rises inside the cell knows that that hormone has done something. The other thing is that there are actual proteins in here that will send a message directly to the nucleus to cause the transcription of a gene. Antibodies. Now, we don't need to know the details of antibodies, we just need to know actually that lymphocytes make them. So you can just write down what you think this is kind of champion knowledge, but we will need to learn it in higher biology. So if you're thinking of doing higher biology, you might want to write all this down and get it in your head. But you'll not be asked too much about the details of antibodies in National 5. Okay? So antibodies are made by lymphocytes. We've got two types of white blood cell in the immune system, lymphocytes and phagocytes. Lymphocytes make antibodies. Phagocytes engulf pathogens. They attack and destroy, seek and destroy cells, right? Whereas lymphocytes, they release antibodies. Antibodies have variable regions that can bind onto pathogens. Now, it's kind of potluck, right? What happens is that the antibody itself has a little region that is just random. The cells are able to make random sites. So these little recognition sites that are on the end of them. And they just put random proteins in, or amino acids in there. And what the purpose of that is if we get an infection, one of those antibodies is likely to have a random site that matches the infection. Matches one of the shape of the bacterium or something, right? And then the system is able to make more of that antibody, right? That, because it kind of remembers. Basically what happens is the cells almost mutate. Right? As lymphocytes divide, they mutate, and each lymphocyte is making a different antibody, right, with a different random site. So if the antibody hits a target, then essentially the message gets back to that cell that made that antibody to divide more. And that cell now starts dividing and making copies of itself that make exactly the same antibody. And suddenly you get an immune response. So lots more of that antibody are made. It's a bit more complicated than that, but you can think of it that way, right? So the message somehow gets back. When that antibody is strike gold, it sends a message back, make more of me, because this is working, all right? And then what the macrophages, one of the things the macrophages are able to do is they're able to see antibodies piled up on something, and they'll go and seek and destroy that as well. Sometimes a lot of pathogens just dodge the phagocytes, sorry. All right? So, different antibodies have different variable regions. Antibodies that attack an infection can be made in vast numbers. We can also have memory cells. So, some of the lymphocytes that are making that antibody will become long-lived cells, right? And they can live for the rest of your life. So that cell that's making that type of antibody will sit around forever. So the only thing that's really important for National 5 learning is that you remember that an antibody is a protein, okay? And it's made by lymphocytes, because that's going to come up again, okay? Membrane proteins. We've already talked about membrane proteins, so this is just a reminder of what we learned. Many proteins are located in the cell membrane to aid transport. There are two main types of membrane proteins. There are channel proteins that make a passage to allow stuff through by diffusion or osmosis. And active transport proteins use energy to move substances against a concentration gradient. Okay, so we've already done that stuff. There's the model of aquaporin at the top, right? Of the protein three-dimensional structure model. All of these amino acids here in the middle are hydrophobic, right? On the outside of it, and they're hydrophilic on the inside of it. So what it means is that the outside of the protein wants to find a hydrophobic area, which is a nice spot in the middle of the membrane is hydrophobic, so it goes and lives there. But the hydrophilic bit in the middle allows water to pass through. Look at them again. Enzymes, this is the big one. Okay, so enzymes are biological catalysts. And this is what we're going to spend the rest of the protein learning on, because they're so important. All right? So a catalyst increases the rate of a chemical reaction. So enzymes allow reactions to happen that would never normally occur at body temperatures. Obviously, we can't heat up the body to get a reaction to happen. Most reactions require a certain amount of energy to get them going, and in nature that would require heat. In physical and chemical systems that would require heat. But you can't put heat into biological systems. So enzymes are able to get those reactions to happen without the use of heat. Sometimes some heat is generated from reactions. Obviously, when we have degradation reactions, heat will be released because we're going from a more complex molecule to a simpler molecule. Generally, that will release energy. But that heat is used by mammals and birds to keep their bodies warm. In fact, we generate a lot of heat in our liver just by burning sugar. The enzymes uselessly burn sugar in order to create heat, and then those products aren't used for energy anymore, they're just broken down from that. Anyway, catalase is an example, breaks down hydrogen peroxide to water and oxygen, which you've been looking at, and it's an example that we need to learn. And amylase is another one. It breaks down, there's me being all Scottish, breaks down starch to simple sugars that can be absorbed by the digestive system. Proteins are large and complicated structures, all right? Some of them are made of thousands of amino acids, all folded together to make a structure, okay? Their function depends on their shape, not their chemistry. They're pretty much made of the same stuff. Amino acids are a little made of carbon, oxygen, and hydrogen, pretty much, with a few bits of other stuff in, sulfur and nitrogen and stuff. But, so at the center of every amino acid, there's a nitrogen atom. their function depends on the shape that they produce. And shapes, if you think that in chemistry, something like platinum is a catalyst, because platinum's attractive to some of the reactants that are in substances, and they all just gather on the surface of the platinum, because platinum has a lot of surface electrons, which is why it's so shiny, right? And it's quite sticky to reactants, which is why platinum is used as a catalyst, because stuff sticks there, it gets them closer together, it gives them more chance of reacting with each other because they're closer together, right? You can think of the proteins as something very similar, except that things kind of get stuck in proteins because the protein shape matches their shape. So they fit a bit like a lock and a key. They come together like interlocking shapes, right? And then things get stuck together. And if that shape is broken, then the interlocking relationship is broken as well. Okay? So structural proteins must hold their shape. Membrane proteins must maintain their hydrophobic and hydrophilic regions. The shape comes from complex folding of the amino acid chain. So here's our amino acid chain up here. It can fold simply because some amino acids will bind to each other. See these? In different ways. They'll have little bits of attraction towards each other. So their primary structure is the primary structure is the chain. The secondary structure is these two possibilities here, the alpha helix or the beta sheet. It's just where the amino acids are a wee bit attracted to each other, and they form flat sheets or spirals, right? The tertiary structure is where they can form they fold right across on top of each other, and various parts of the amino acid can join up. And this is done with the help of other proteins and enzymes in the cell. And it's folded into a big globule or whatever, and you'll have spirally regions and sheety regions and all sorts. But you make a shape, okay? And the quaternary structure, this is where other things are incorporated into the structure of the protein. So it could be other proteins. Many proteins are made by several individual genes, products getting together and making a single big protein. That means you're taking several different genes, several different proteins, putting them together. That's the quaternary structure. Or you, in this particular group, we've got the heme group, which in hemoglobin, the heme group is incorporated into the structure of the hemoglobin protein, right? But the shape is held together by delicate attraction between different amino acids, and that delicate attraction is very easily disrupted by heat, which is why you can't heat up biological systems. Okay? That's a lot to write down. See if you can get it down, and then we'll stop. Okay? Good. So the last thing we need to learn about this is about denaturation. So basically that's what happens when you heat a protein up. It loses those fine connections, those very loose bonds that hold the protein together. As long as the protein's fine, as long as it's got the right conditions. But if it gets hot, or it in any way, temperature causes molecules to move faster. It breaks those bonds, right? And you can see this with an egg. And the other thing is that it's irreversible. Once those bonds are broken, there is no system in the cell to put it back together again. The protein is just broken down and lost in the cell, or that's if the cell survives the heat shock. Or in the case of an egg here, where the albumin is a protein, albumin is a protein that's put into eggs. When we heat it up, it turns white. That's irreversible. When you cool it down, it doesn't go clear again, right? So that's a good example of denaturation of a protein.